Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 12th January 2021. As we have been revising the entire syllabus in a row, as part of it we are revising in the form of important questions in the chapter. So here in every model question paper or test paper, there will be 15 questions consisting of multiple choice questions, 1 mark questions, 2 mark questions, 3 mark questions as well as 5 mark questions. So once we perform, let's say 2 to 3 papers, you will have an idea in which concept of the chapter maximum marks are coming and from which concept objective type questions are coming and descriptive type questions are coming. So you can have a self-analysis of the entire chapter and you can make a short notes, personal notes for each chapter like this, so that out of the 11 chapters that are going to be there for your batch, what are the concepts that you need to focus on? Maybe something like 50 concepts, so that you get 70 out of 70. That is the target. So with that in our mind, with that ambition in our mind, let's go the first question. State the use of molecular medicine. Now keep a paper and a pen with you. Start answering the questions. At the end of you finishing that, switch over to the answer session that I am going to give. Have self-verification. Okay. State the use of molecular medicine. A. Used to understand several diseases like Alzheimer's, Parkinson's diseases. B. Used as gene therapy. C. Improves diagnosis of the diseases. And D. All of this. Of course, molecular medicine, I think all of this, diagnosis of the disease, gene therapy and all that. Okay. So I think answer should be D. Question number 2. DNA fingerprinting or genetic fingerprinting is the process of matching the fingerprint of different persons, analyzing VNTR samples of DNA obtained from body fluids or cells. I think that should be the answer. C. Finding similarities of finger shapes of different individuals. Not at all. D. Comparing the DNA of two individuals. I think it is VNTRs, variable number of tandem repeats. Question number 3. DNA probes used in fingerprinting are highly sensitive electron microscope, UV beams, X-ray scanners, DNA segments having radioactive isotopes. That should be the answer. Question 4. Best method to determine paternity is parental, is protein analysis, gene counting, chromosome counting. Of course, just now we said DNA fingerprinting. That should be the answer. And 5th, a gene of operon which synthesizes a repressor protein is structural gene, regulator gene, operator gene, promoter gene. It has to be regulator gene. Now, these are the multiple choice questions. Now we go for the objective type questions. What is the distance between two base pairs in DNA? Straight knowledge level question, just memory based one. Question number 7. Refer to the following diagram and answer the given questions. Only one question is there. So here, what type of bond is represented by dotted lines? These lines which are there, what type of bonds are there? That is the thing. So here you understand that this is DNA molecule, S for sugar, B for base pair and the phosphate group. So what is the bond? What type of bond exists between sugar and base pairs? That is the question. Next, question number 8. Corona and his colleagues synthesized an RNA molecule with repeating sequences of AG nitrogen bases. That is AG, okay. It produced a tetrapeptide with the alternating sequence of valine and tyrosine. What are valine and tyrosine? Amino acids. It proves that codon for valine and tyrosine are, okay, what are the codons? Question number 9. Name the types of synthesis. A, that is this, A synthesis, and B synthesis occurring in the replicating fork of DNA as shown below. Next, question number 10. What is teminism? Repeatedly this question is asked. Important question. Question number 11. Mention two functions of codon AUG. Of course, you know it is initiating codon, August, AUG. 12. Differentiate between the process of transcription in prokaryotes as well as transcription in eukaryotes. Difference between that. 13. A couple quarreled with the hospital authority on suspicion that their child had been exchanged after birth. The couple based their argument on the fact that their child is O blood group, whereas they are A and B blood groups respectively. The doctor smiled and explained. 1. What values of the doctor is reflected here? Question 2. How can the child be O blood group as explained by the doctor? Next, which test method can be considered authentic to identify the biological parents of the child? 4. Name the other blood groups which the child could have inherited. So it's a beautiful question. This is case study question. This is the new type of question. So you need to have a lot of practice. Okay. Question number 14. Of course, question number 13 is related to genotype. Once you know that multiple alleles, okay, you are thorough with that multiple alleles, you can easily answer this question number 13. Question number 14. Differentiate between leading strand and lagging strand. You are drawing straight line and then dotted line. Leading and lagging. Next, very important experiment. How did Hershey and Chase prove that DNA is the hereditary material? Very important question. Number of times got repeated in the board exam. Explain their experiment with suitable diagrams. Draw beautiful diagrams for that. Then you can explain. So let's go for the answers now. Answer session. Question number 1, as I said, all of these. That is, the application of the power of molecular genetics to the problem of human disease plays an important role in many of the research programs in the Department of Biology. Several complementary approaches are used by our research groups. The power of genomic analysis is used to identify, isolate, and characterize genes which cause and contribute to the etiology of human diseases. Human disease is also studied through the functional analysis of the key genes. Low density lipoprotein receptors in arteriosclerosis and stroke, broad spectrum of tumor suppressors and oncogenes in cancer, genes directly leading to the disease etiology in neuromuscular disorders such as Alzheimer's, Huntington's disease and muscular dystrophies. That's why the answer should be molecular diagnosis covering entire thing. Question 2, of course, just now I said VNTR, variable number of tandem repeats, analyzing VNTR samples of DNA obtained from body fluids or cells. So VNTR analysis, sometimes it is called DNA fingerprinting, can be used to identify and match cell samples. In order to use VNTRs, we must have a source of DNA. The DNA can come from any nucleated cell. White blood cells, skin samples, semen samples, hair follicles. Red blood cells cannot be used as they do not have a nucleus. For a VNTR analysis, the DNA is extracted and cut using restriction endonuclease, which are molecular scissors we call it. Southern blot is then performed using various probes that can be repeated. Question 3. DNA segments having radioactive isotopes. Explanation: in fingerprinting, the nylon membrane was incubated with radioactive probes. DNA probes are small fragments of mini satellite DNA tagged with radioactive phosphorus. This is what the definition of DNA probe. The probes only attach to the pieces of DNA that they are complementary to. In this case, they attach to the mini satellites in the genome. Radioactive isotopes can be easily identified using scanner. Question number 4. DNA fingerprinting. DNA paternity testing is the most accurate form of paternity testing possible. If DNA patterns between the child and the alleged father do not match on 2 or more DNA probes, then the alleged father can be totally ruled out. If the DNA patterns between mother, child and the alleged father match on every DNA probe, the likelihood of patent is 99.9%. That is the accuracy. Question number 5. Regulatory gene. This is regulatory gene is a gene that regulates the expression of one or more structural genes by controlling the production of a protein such as genetic repressor, which regulates the rate of transcription. Regulation of lac operon by repressor is called negative regulation. So you need to draw the diagrams, both the diagrams, lac operon positive feedback, lac operon negative feedback. Then you can understand this concept clearly. Question number 6. I said knowledge based question. The diameter of DNA is 20 angstroms and the distance between base pairs is 3.4 angstroms. The base pairing of opposite strands is stereochemically selective. Adenine always pairs with thymine, so as guanine always pairs with cytosine. So only we always write A for adenine with a thymine, guanine and cytosine. Of course, between adenine and thymine there are double bonds, guanine and cytosine, triple bond, weak hydrogen bonds will be there, that can be broken. That's how the DNA replication takes place. And seventh one, of course, hydrogen bonding. This bonding is hydrogen bonding, just now I said. Okay. And then question number 8, codon for valine AGA, the triple codon for tyrosine GAG. And the question number 9, A, synthesis of leading or continuous strand, leading strand, and B, synthesis of lagging or discontinuous strand. I said if there is a straight line like this, that's a continuous. If there is breaks like this, then we say lagging strand or discontinuous strand. Question number 10, reverse transcription is known as teminism, reverse transcription. So this was first reported by a scientist called Temin and Baltimore. That's why the name teminism. RNA of some viruses like retroviruses first synthesize DNA through reverse transcription. Normally, what is transcription? The DNA becoming RNA. DNA replicates, breaks and forms RNA. Now this is reverse transcription. That's why. So the DNA then transfers information to RNA, which takes part in translation of coded information to form polypeptide. Question number 11, it codes for methionine amino acid, that is initiating codon, AUG, functions of AUG, initiating codon. It codes for methionine amino acid, say MET, methionine. It acts as initiator codon for protein synthesis. Next, difference between transcription in prokaryotes and transcription in eukaryotes. Production of transcription becomes effective in situ. Production of transcription come out of the nucleus for functioning in cytoplasm. There is only one RNA polymerase. 3 RNA polymerases takes part. mRNA is polycistronic. mRNA is monocistronic. Splicing is not required. Splicing is required for removing introns. Now, question number 13, which I said case study type. Very, very important question. You need to train lot of questions in this. The doctor was assertive, patient, and pragmatic. These are the qualities, okay, that he exhibited. Question 2. It is possible for if the parents are heterozygotes, that is AI, that is BI, that is A0, I0, B0, I0. If the child receives I0 from both the parents, it becomes I0, I0 and expresses O blood group. Very much possibility is there. Like the chart, we can see like parent A, blood type A, parent B A, then the genotype. In fact, what is missing here is the genotype. You should write when A is, then it has to be IA or I0. Two choices are there, I0. Similarly, this case. So, if it is I0, then blood O group also possible very much. So, these are the various formulations that you need to understand so that we can come to know what are the possibilities of the child who is going to take birth with the heterozygous or homozygous blood groups of the parents with the RH, of course, RH positive, RH negative. So there can be RH positive and RH negative, but if both the parents are RH negative, then the child has no option, has to be RH negative. Next, DNA fingerprinting is the only solution to find out, and it can be, as we said, it is not necessarily O, it can be A as well as B blood group. Here comes this one, this combination. So there can be A, B or AB. So any chances are there. Next, so this is what we are doing it. The parent's blood group is A, let us say. Then the genotype will be IA, I0. Another parent blood group, let's say mother's is B, then the genotype is IB, I0. So during this meiosis, when the gametes get separated, the gamete will get one gamete IA, another one will get I0 gene, IA and I0. This has to be IB, not A, B, it has to be B. So when fertilization takes place, gametes fuse. What are the possibilities? One side you got Punnett square IA, I0, another side IB, I0. So the combination of these two, IB, IB. So you get IA, IB. Then this and this, IA, I0, I0, IB, and I0, I0. So what are the phenotypes of this? IA, IB, the blood group has to be AB. Baby's blood group will be AB. If it is combination of IA, I0, it has to be A because A is dominant over not having any antigen. IB, I0 again B is dominant, and if there are no antigens at all, it has to be O group, I0, I0. Question number 14. Leading strand and lagging strand differences. Leading strand, it is a replicated strand of DNA which grows continuously without any gap. The lagging strand is a replicated strand of DNA which is formed in short segments called discontinuous strand. Leading strand, it does not require DNA ligase for its growth, whereas DNA ligase is required for joining Okazaki fragments, that pieces. Recollect from your memory these pieces discovered by a Japanese scientist Okazaki, so they are named as Okazaki fragments. The direction of growth of a leading strand is 5' to 3', whereas direction of the lagging strand is opposite, 3' to 5'. Only a single RNA primer is required. Starting of each Okazaki fragment requires new RNA. Its template opens in 3' to 5' direction, opposite. Its template opens 5' to 3' direction. Formation of leading strand begins immediately at the beginning of replication. Formation of lagging strand begins a bit later than that of leading strand. Next, Hershey and Chase experiment. Chase experiment 1952. Hershey and Chase conducted their experiments on the T2 phase, a virus. The phase consists only a protein shell containing its genetic material. In the first experiment, they labeled the DNA of phases with radioactive phosphorus P32, the element phosphorus is present in DNA but not present in any of the 20 amino acids from which proteins are made. They allowed the phages to infect E. coli, then removed the protein shells from the infected cells with a blender and separated the cells, and viral coats by using a centrifuge. They found that radioactive tracer was visible only in the pellet of bacterial cells and not in the supernatant containing protein cells. In second experiment, they labeled the phages with radioactive sulfur 35 after separation. The radioactive tracer then was found in the protein cells but not in the infected bacteria, supporting the hypothesis that genetic material which infects the bacteria is DNA only. So you need to draw this diagram and explain each phase of this. Then you have a clarity on the concept. So this is how you need to answer specific and drawing diagram.